Welcome back to our fifth lesson and our fourth lesson recapping cellular processes. We'll be talking about homeostasis and the cell membrane and how molecules move in and out of cells. Homeostasis is being able to maintain a stable internal environment despite any external factors that are happening on the outside. I like to compare homeostasis to your thermostat in your home. You set your thermostat to a specific set point. Maybe it's 72 degrees Fahrenheit. And we live in sunny Arizona and it's 114 degrees outside. And that heat is starting to influence the internal factors of your home. It's starting to get warmer. That increase in temperature will be detected by your thermostat. Your thermostat will then turn on your air conditioning unit that will then cool down your home back to your set point. In turn, when it's chilly outside, maybe it's one of these cold January 45, 50 degree winter mornings, and that cools your house down. The thermostat will also detect that change, turn on your heater, and raise the temperature back up to its set point of 72 degrees. So we have to stay within homeostasis. We have to keep relatively narrow limits within the cell. Otherwise, the cell cannot function correctly, and we start to see disorders and diseases from the cell not functioning properly. So in order to stay stable on the inside, the cell has to be able to regulate things moving into the cell and moving out of the cell. What's going to create a barrier is the cell membrane. And that cell membrane will control what can go into the cell and what can go out of the cell. So what creates this barrier is these two layers of phospholipids. The cell membrane is said to be a phospholipid bilayer. So I have one layer of these phospholipids and a second layer. These phospholipids have a shape. There is a head made out of a phosphate group that is really, really, really hydrophilic, meaning it is attracted to water. And then there are these hydrophobic tails. There are two hydrophobic tails. And those are water fearing, meaning they really do not like water. So these two layers will form because those hydrophobic tails will join together because they're off. They want nothing to do with water. So they're going to hang out together, and the hydrophilic heads are going to interact with the water so those hydrophobic tails can stay on the inside. So that is pretty convenient because the inside of the cell is a pretty watery environment, and so is the fluid that surrounds our cells. There's this extracellular environment, and that extracellular environment is filled with this interstitial fluid that is very watery. So this cell membrane is specialized, and it's said to be semi-permeable because only some things are going to be able to pass through this phospholipid bilayer. Other things will not. So if anything has a charge to it, it's going to get rejected. It will not be able to enter into that cell. It won't be able to go past that phospholipid bilayer. Also, if the substance is too large, like glucose is actually a really large molecule. Oh, wow, that's a horrible glucose molecule. Pretend like that didn't happen. So glucose is too large to pass through that cell membrane. But the cell needs things like water. Water is polar, and polar molecules can't pass through that membrane. So the cell membrane has things like aquaporins, which are protein carrier channels that allow water to freely move in and out of that cell, which also means that the environment that the cell is placed in could have a very drastic effect because water will move freely through these aquaporins, which we'll talk about later. The cell membrane will also have carrier proteins for things like glucose that can't get through that cell membrane. They will also have glycoproteins and carbohydrates on the surface that can act as receptors and for cell identification. Those little receptors also help the cell respond to its environment. Before we start talking about how molecules move in and out of cells, we need to recap some chemistry. Recall matter and energy tend to flow down concentration gradients. A gradient is a difference in either a chemical concentration, a charge being positive or negative, a temperature, or some kind of pressure between 2 points. We talk a lot about this in 202 when we talk about blood flow and blood flow through the heart. Blood flows down a pressure gradient from an area of high pressure to an area of lower pressure. When we talk about action potentials, these chemicals flow down a concentration gradient. Electrical gradients are when charged particles flow down an electrical gradient. And you can even have an electrochemical gradient where there's a combination of the concentration and electrical gradient. And heat will flow down a thermal gradient. Movement in the opposite direction typically requires energy. While when molecules move down their gradient, that's typically a passive process and it's going to just happen given enough time. While if we try to go against the gradient, that's like swimming up against the current or trying to swim upstream. It's going to require energy to move upstream versus if you just jump into a tube and you're going down the lazy river. Right? Or I don't know if you guys are too young to have ever had a red flyer wagon. We used to have these little wagons when we were young, and we would pull them up a hill, which would require a tremendous amount of work. But it was usually worth the reward because we would just sit in that wagon and we would travel down without any energy. Here we have a diagram of how there is flow down gradients within the cardiovascular system. The heart is a pump and it will create a large pressure inside of the heart as it contracts. And there'll be a low pressure in the arteries. So blood, as pressure builds, will have no choice but to go out of the heart and travel down their pressure gradient to the capillaries within our tissues that have lower pressure. This also happens when we eat. As you digest your food, your body will begin to break down that food into its building blocks or monomers. So when you eat complex carbohydrates and starches, your body will break that down into its individual monomer called a monosaccharide or glucose. And so there will be a high concentration of glucose within the lumen of your stomach and a low concentration of glucose inside of your intestinal cells. So by a passive transport, that dietary glucose will just flow down its chemical gradient into the cell to be absorbed and then into your blood and carried out through your tissues to be used. Same thing goes for ions like sodium or potassium. Ions will flow down their concentration gradients. And this is going to have a big play when we talk about it in the nervous system. And then same thing with how our bodies actually cool down. When you get warm, your body will start to dilate the blood vessels and direct blood vessels to your skin. So when you exercise, sometimes you get red. Well, that warm blood is now in your skin, and heat will be able to flow down its thermal gradient to the environment and cool down your body. So I keep mentioning that these molecules, if given enough time or if they move down their gradient, it's a form of passive transport. So what passive transport means is that the molecules will move naturally from where there's a lot of them or a high concentration to an area where there is a low concentration. And this will just happen over time. It does not require any energy. So when water moves, water will move through this process of passive transport. But the movement of water has a very special name. It's called osmosis. While the movement of any other substance down its concentration gradient is known as diffusion. What facilitated diffusion is sometimes those molecules just are too big to get through that cell membrane on their own. So larger molecules like glucose or charged molecules like water will need to be helped or facilitated across the cell membrane by channels. And those channels are made up of proteins. They still will move from an area of high concentration to an area of low concentration. They just need a little bit of help to get through that cell membrane. So this is called facilitated diffusion. So when areas move from an area of high concentration to an area of low concentration, no energy is going to be required as those molecules move through the cell membrane. And they're going to continue to move until dynamic equilibrium is reached. So what that means is if you think about equilibrium, think of equal. There's an equal amount of molecules on each side of that cell membrane. But it's not that the molecules aren't moving. They're still moving around. They're still going in and out. It's just they're going in at the same rate that they're going out. So there's still the same amount inside and outside of that cell. So this is how gas exchange works in the capillaries of your lungs and in the capillaries of your body tissues. That capillary wall, we'll talk about what lines it, what type of epithelial tissue, but these molecules will have to move through the lining of your lungs and the lining of your tissues. So larger molecules might not be able to pass through that capillary wall, but smaller molecules will be able to pass through that membrane of the cell that makes up the lining. So when you breathe in air, there's a high concentration of oxygen in the alveoli of your lungs, and there'll be a low concentration. While there'll be a high concentration of carbon dioxide on the inside and a low concentration of carbon dioxide in the alveoli of your lungs. So by this process of diffusion, those carbon dioxide molecules will move from where there's a high concentration on the inside of your blood to the low concentration in your lungs, and the oxygen will move from a high concentration inside of the alveoli to the low concentration of your blood, and we'll have that gas exchange happen. Because our cell membrane is permeable to water, meaning water has those aquaporins and water can freely move in and out of the cell. This means the solution that your cells are placed in can have a drastic effect on this cell. So let's look at the red blood cell. When you are putting a patient on an IV, that solution is isotonic to the red blood cell. What that means, iso means the same, tonic, think of strength or strength of concentration. So that saline solution has the same amount of salt as your red blood cells. So water is attracted to salt, and water will follow salt. So if there is more salt inside of the cell, or if there's salt outside of the cell, water will follow it. Salt sucks. Right? So if the red blood cell is placed into an isotonic solution like saline, that means water will diffuse in and out of that red blood cell equally. But if you happen to put that red blood cell in salt water or a hypertonic solution, that means that that solution has more salt or solutes than are inside of that red blood cell. So all of the water will leave that red blood cell, and that will cause the red blood cell to begin to shrivel or be cremated, right? Water will diffuse out. And if that red blood cell is not fixed, the water doesn't go back into it, it will eventually die. And you might be thinking, well, why don't I just use pure water, like some distilled pure H2O? Well, that is called a hypotonic solution. So in that case, there's more salt on the inside of the red blood cell. So water will move into the red blood cell because the salt inside of that red blood cell will suck the water into it, right? Salt sucks. So the water will move to where that salt is and go into the red blood cell, and it will cause that red blood cell to swell. It will eventually burst or lyse, and that can cause death. Sometimes a cell needs to have something even though there is more of that substance inside of the cell. Or sometimes we need to move a molecule somewhere where there's already a high concentration of it. For example, when you learned about cellular respiration, you learned that glucose is moved into the mitochondria of the cell to perform cellular respiration. And there's already a high concentration of glucose inside of that mitochondria. So we're going to use what's called active transport. Active transport moves molecules from an area of low concentration to an area of high concentration. So this is the opposite than the natural process. So the cell does have to use some energy and put in some work. It will require energy. And there are two main types of active transport. We have endocytosis, right? If you break down that word, endo means within, cyto means cell, cis is process of. So that's the process of moving into the cell. Where exocytosis, exo means out or exit. So this is the process of moving out of the cell. And both of those types of active transport will require energy. So they're going to use what's called carrier proteins. And we're going to talk about carrier proteins a lot when we talk about nerve conductions. So these guys will move molecules against their concentration gradient. And we'll talk about the necessary functions for life when we come back.